So my name is William Oakes, and together with Clea Hoxton and Nicole Ward, we are research assistants in the laboratory of Drs. Brian Lee and Gabriela Perez Alvarado, PhDs. We're looking at structural and functional investigations into the regulatory roles of small RNAs in the Streptococcus genus. A little bit about why we're doing what we're doing. Antibiotic resistance is a growing problem. The CDC estimates more than 2.8 million antibiotic resistant infections per year in the U.S. alone, with 35,000 patients lost. Of those infections, between 11 and 24,000 are streptococcal, with between 1,200 and 1,900 patients being lost. So needless to say, there are new and novel antimicrobial solutions needed. A little bit about the Streptococcus genus. Strepto is from the Greek meaning twisted and coccus meaning sphere. Strep divides along a single axis, which gives rise to pairing, causing these chains that you see depicted in the figure to the right. Strep was the first microbe to be linked to contagious disease. Pasteur was investigating postpartum puerperal fever and noted that he found strings, quote, strings of beads in the infected patients. This, of course, turned out to be strep and led to aseptic technique in hospitals. Strep is a gram-positive bacterium. The peptidoglycan in the cell wall absorbs the gram stain, so we refer to it as gram-positive. It's catalase negative, that is, it lacks the enzyme that converts hydrogen peroxide to oxygen and water. It's a facultative anaerobe, meaning that it will utilize aerobic respiration if it has oxygen handy. If it doesn't, it will switch to fermentation. A little bit about the pathogenicity and some of the bad actors. Strep pyogenes causes everything from strep throat to necrotizing fasciitis. Pneumoniae causes pneumonia. Agalactae, a problem with meningitis and sepsis in neonates. Dysgalactae causes meningitis, swelling in the sac around the heart, bacteria in the blood. So a lot of bad actors. Bad stuff there. So a little bit about our approach. It's pretty common knowledge that DNA is copied or transcribed into single-stranded RNA, which is then translated into protein. The figure down at the bottom, the light blue circle, represents RNA polymerase, which is an enzyme that attaches and travels along the DNA, double-stranded DNA. It essentially catalyzes reactions between complementary bases on the DNA and incoming nucleotides, resulting in the multicolored chain that you see in the figure there, labeled RNA. Not all of that RNA, however, is mRNA. That is, not all of it is translated into protein via mRNA, and it is these RNAs that we are interested in looking at here. DNA regions which correspond directly to mRNA, however, are referred to as coding regions. The regions of DNA which do not correspond to coding regions are known as intragenic regions, and we now know that these intragenic regions contain instructions for small RNAs with extensive regulatory functionality. So I want to shift gears up from the transcription gene level now and talk about what the gene that we're looking at, or at least the area that we're looking at preceding this gene, what this gene actually translates to. The protein is a protein with enzymatic activity called aspartate semialdehyde dehydrogenase. This reaction, although it is reversible and commonly written in this direction, starting with the aldehyde and moving to the aspartyl phosphate, generally under physiological conditions it would run in the opposite direction. That is, that the enzyme via a thiol at the active site would cleave that ester linkage to the phosphate and then facilitate reduction with the help of NADP on down to the aldehyde, which is the structure that you see there on the left. Um, this semialdehyde is an immediate precursor for dihydrodipicolinate, which is implicated in gram-positive sporulation, in particular Clostridium difficile. C. diff is a particularly sometimes impossible bacterial infection to treat because of sporulation, its capability to sporulate, which allows it to evade the immune system. That aldehyde is also implicated in lysine biosynthesis, which is immediately implicated in bacterial cell wall cross-linking. It's also an immediate precursor to homoserine, which is converted to methionine and to S-adenosylmethionine, which is implicated in many methylations, including DNA methylation. It is also implicated for bacterial quorum sensing, which is the regulations of genes in response to the cell's own population. A little bit more about streptococcal small RNA. These RNAs are relatively short. The one we're looking at S-thermo is 151 nucleotides. They are generally between 500 and 300 nucleotides in length. They are non-coding, as I mentioned, that is, they are not translated to protein. The mechanism by which they operate, perform transcriptional and translation gene regulation, is by interaction with mRNA. They can also prevent protein interaction by sequestering molecules from proteins, and also by interaction with cis elements, meaning that they can regulate or interact with the same gene within which they are encoded to promote or inhibit the expression of that gene. This is part of our working hypothesis, is that this small RNA that we're investigating acts as a ribose switch. That is, it binds to the mRNA to block access to the ribosome or not block it, depending on some extracellular stimuli. Non-covalent interactions of the bases of these small RNAs give rise to folding into very structured stem loops, which dictate the functionality of the small RNA. We chose S. thermophilus, particularly ATCC strain 19258. This is because it allows access to people, students at our level, undergraduate and high school. Because it is not pathogenic and safe to work with, doesn't require extensive training, also because it has probiotic and commensal activity, also because it has significant evolutionary conservation across the genus as a whole, and also at the 5' untranslated region of the ASD gene that we're looking at. You can see the similarities in the structures to the right. This is S-thermo, sRNA, secondary prediction at the top. On the top, S mutans at the bottom left, and S pyogenes at the right. The two on the bottom are both pathogenic. I'd also like to mention that we named these stem loops because they appear in just about every Streptococcus species we looked at. The first loop we refer to as the P1 loop. P2 through P5 we refer to as the clover leaf, and P6 as the terminator. As I mentioned, S-thermo is 151 nucleotides long for the sRNA. The sRNA for mutants is 139. So a little bit about searching for targets of the ASD sRNA. The figure on the left is, again, a secondary structural prediction for the sRNA via RNA fold. Copra RNA is a software package from Freiburg University, which uses complementarity of base pairing of different related genomes across, for example, different databases, NCBI and others, to predict interactions based on this base pairing, these base pairing interactions. And the x-axis represents increasing nucleotide, increasing length, and therefore more nucleotides. So you can see there's quite a lot of predicted interaction around the P1 loop, quite a bit around the cloverleaf structure, and quite a bit around the terminator region. Also interesting is that the Copra RNA also makes annotations of predicted proteins that could likely be involved, and there were quite a lot of ribosomal proteins predicted. Also, Clea mentioned that some were implicated in biofilm formation as well. So a little more about the P1 stem loop in particular. Non-covalent interactions, as I mentioned, between these nucleotides determines their secondary structure, and that in turn determines their three-dimensional structure. So structure, of course, the three-dimensional structure, of course, determines the function of the sRNA, which is ultimately what we'd like to get at. In fact, we'd eventually like to get to the point where we use X-ray crystallography or NMR to get quite a lot of confidence about what the three-dimensional structure looks like. But the fact that secondary structure dictates three-dimensional structure allows for an additivity of analysis to where we could separately analyze these structures, the P1, the cloverleaf, and the terminator regions, and still get pretty accurate prediction as to what, in other words, our analysis were additive. It allowed our analysis to be additive. So a little bit about the DNA corresponding to the ASD sRNA. We prepared M17 broth and agars, which were prepared with 0.5% lactose. Streptococcus thermophilus was cultivated under anaerobic conditions, and the genomic DNA was extracted via the Promega DNA extraction kit. We designed primers and isolated and amplified the DNA region for the full-length structure of the ASD sRNA via PCR, also for the cloverleaf and terminator structures. In the gel that you see to the right, we ran a 2% agarose gel. Lanes 4 and 5 represent the full-length region, which is around 151 nucleotides, as you can see. The P1 and cloverleaf regions are a little bit smaller, and lanes 8 through 11 represent the terminator regions, which came back a little bit smaller still. This is a gel that we did to test T7 polymerase toward producing the ASDS RNA. RNA polymerase catalyzes the formation of single-stranded RNA from double-stranded DNA. It requires a magnesium cofactor for synthesis. T7 polymerase is very promoter specific. Ideally, it looks for triple G AGA. The lanes 3 through 5 here in this gel began with transcripts beginning with just AUG and didn't produce much. Lanes 8, 9, and 10, we added a single gene nucleotide for GAUG. It did a little bit better. Lanes 12, 13, and 14, we added GGA for GGA AUG and got a better result, although the bands underneath the smaller bands we think are attributed to abortive transcripts, as T7 polymerase tends to fall off and reattach. But the larger structures do seem to be about where we would expect them to be. So I'd like to close with about computational prediction of three-dimensional models. Three-dimensional, structure, as I mentioned before, will elucidate regulatory activity, which is what we'd like to get at. We use the FAR2 software by the DOS laboratory at Stanford, which essentially starts with small known fragments and increases probabilistic optimization of energy functions. It's very computationally expensive because it's a Monte Carlo process, but we do have the program now on the high performance cluster here at Coastal. The prediction that you see there is the P1 stem loop from the web server. It also does a statistical analysis for you, which gives you a root mean distance a resolution. This one came back at about 7.05 angstrom. This is the distance between the best structure and the ones closest to it. But in the future, we will be able to carry out those predictions locally, which is pretty exciting. So we'd like to say thank you to the South Carolina Embry, to the National Institute of Health, General Medical Sciences. Um, Thanks a lot to Dr. Wakefield for putting everything together this summer. Thanks a lot to Dr. Richardson for sharing his laboratory with us, putting up with us. Thanks a lot, of course, to Dr. Lee as always, and Dr. Perez Alvarado for everything they've taught us this summer. Thanks as always to Coastal Carolina University, no place like home. And thanks, of course, to Clea and Nicole.